All right, thanks for coming, everyone. My name is Ryan Schott, and I'm going to be talking about the evolution of frog vision today, and in particular the roles that ecology and ontogeny play in visual systems of frogs. All right, if I can get it to advance. All right, so frogs. I'm sure everyone's familiar with frogs or anurans. Them, along with the other amphibians, are the sister group to amniotes, and it's really species-rich clade. But why are they interesting for studies of visual evolution? Well, they have this species richness, as I mentioned. They also represent over 200 million years of evolution, as well as many diverse ecologies, life histories, and behaviors. As this combination provides a lot of landscape for visual adaptation. And today we're going to focus on two aspects of this. We're going to focus on their different habitats, as well as their different life histories. So most frog species are biphasic, meaning they transition from a larval tadpole stage to an adult stage. And typically this transition is from an aquatic tadpole to a terrestrial or semi-terrestrial adult. But of course, some species are aquatic, like you can see in the blue frog on the bottom there, and they never leave the water. And so this biphasic life cycle is common for many frog species, but there are others that are direct developers, meaning they develop directly within the egg into a adult morphology, just a tiny version of it. And so this diversity of habitats and behaviors, life histories, all of that is providing a lot of different light environments that frogs utilize. And so our kind of general hypothesis here is that these different light environments will have imposed different selective pressures on frog visual systems, resulting in different visual adaptations. And my colleagues and I have been studying this, taking a kind of comprehensive view, combining spectral analyses, molecular evolution, and morphology. And today we're really going to focus on the habitat and ontogeny or life history side of things. So here I just want to acknowledge off the bat my collaborators on this paper. This is really only a small subset of our kind of collaborative group. Really, we have a global network going. So these are a large group of people that have contributed many different things to these various projects, whether it be field work, collecting specimens, access to museum collections, or expertise. All right, today I'm going to focus on three aspects of frog vision, looking at the frog eye, starting with overall eye size, and then looking at the lens and briefly at the retina. And for the lens, we're going to talk about lens transmission and shape. And then within the retina, or molecular side of things, we're going to look at eye and retinal gene expression and evolution. All right, so starting off with eye size. Why is it important? Well, eye size helps to provide the sensitivity and the acuity of the eye. So a larger eye is going to result in higher absolute sensitivity, meaning less light is needed to elicit a signal, as well as higher acuity, meaning the resulting image produced will be sharper or higher resolution. Now, a smaller eye is going to result in lower sensitivity and lower acuity. Now, of course, all animals don't just try to evolve the largest eye possible. There's going to be some trade-offs there in terms of developmental and metabolic costs, as well as susceptibility to damage. All right, so how do we study eye size across frogs? We use this extensive phylogenetic sampling, and we looked at eye allometry, meaning the size of the eye compared to the body size. This is just an example of the level of sampling we have. I'm expected to be able to see anything specific on this figure, just to show you the broad sampling that we used. So one of the first things we noticed in this study is that frogs have relatively large eyes for vertebrates. So the green circles there on the green line are the frogs, and they have these larger eyes than most other vertebrates that we looked at, so birds, fish, and mammals. And so we think this is providing good evidence that vision is really important for frogs, despite the fact that most frog species are nocturnal. But how does habitat influence eye size? We're really going to focus on three of the groups here today: scansorial or tree-climbing frogs, the aquatic frogs, and the fossorial frogs, which spend most of their life cycle underground in a subterranean environment. And so some of the things that popped out from these results is that scansorial frogs have relatively large eyes. And so remember, this is likely to provide higher visual acuity, whereas the opposite was true for the aquatic and fossorial frogs. They have relatively small eyes, and this should provide lower visual acuity. But what about looking across ontogeny? And so we did this in a smaller subset of frog species, so we don't have as extensive a phylogenetic sample, but you can see there from the blue dots and blue lines for tadpoles and the green dots and green lines for the adults that across species, the allometric slopes, so the rate of eye growth, differs considerably across these different species. And so one of the things that popped out from these results is that the tadpoles of aquatic and fossorial frogs as adults have lower allometric slopes, meaning these tadpoles are investing less into eye growth. Now, the same is not true of the adult slopes in this case. All right, so switching to looking at the lens, starting with lens transmission. Why is this important? In order for light to be detected, it has to first pass through the lens to reach the retina. And the lens is not necessarily transparent equally to all different wavelengths of light. So on this graph, you're going to see on the y-axis the level of transmission of light at different wavelengths, going from UV to the near infrared. And so we did this for a number of different species of frogs, a subset of which is shown here. And this is the UV range of the light spectrum. And so frogs in this area have high transmission of UV light, meaning the UV light is reaching the retina and could be detected. Whereas frogs in this area are blocking UV light. The UV light is being filtered out and it's not reaching the retina. And so one of the things that popped out from these analyses is that scansorial frogs tend to have increased UV filtering. So they're blocking UV light. And we think that this is likely to reduce in higher visual acuity because shortwave light tends to scatter and can have chromatic aberrations, which would reduce the clarity of the resulting image. Now, we also measured lens shape using high-resolution CT data in both tadpoles and adults. And when we looked at this across our different habitats, we found that aquatic and fossorial frogs tend to have spherical lenses like the tadpoles did. So all the tadpoles in our sample had spherical lenses. This is typically expected in aquatic vertebrates because the spherical lens in water will produce a sharp image, whereas to produce a sharp image in air, you need a flattened lens. So most frogs, as they transition from a larval tadpole to an adult, flatten their lens, but we found this was not the case for aquatic and fossorial frogs. Not surprising for aquatic, but a bit unusual for fossorial frogs because this suggests that they may be myopic or nearsighted. But perhaps this is not a big deal for these species that are living underground and they aren't using their distance vision. We also found that aquatic frogs had relatively small lenses compared to other frog species from other types of habitats. So again, suggesting perhaps they're investing less in their eye and in vision generally. All right, so switching to the molecular side of things, we're going to look at eye and retinal gene expression and evolution, starting with changes in the eye transcriptome across ontogeny. And so we studied this across the whole transcriptome of the eye. And so in this volcano plot here, the red dots on the left side represent genes in the eye transcriptome that are upregulated in tadpoles, whereas those on the right side are genes that are upregulated in juveniles. And we found that a really high percentage of genes in the overall transcriptome was differentially expressed. And of those genes, a high proportion of those were actually visual genes. And so in this volcano plot, this is a different analysis where we just restricted the data to a subset of curated visual genes. And we found that several gene types popped out from this analysis. So we had the opsins, the lens crystallins, which are involved in the formation of the lens, the phototransduction genes, and a gene involved in chromophore function. All right, but what about across species? To do that so far, we've focused in on the retina and in particular the rod and cone photoreceptors and the light-sensitive visual pigments that they contain. So these are the molecules that are in our eye that actually detect and respond to light, and they're composed of an opsin protein and a vitamin A-based retinal chromophore. And ancestrally in vertebrates, we have 5 different types of opsin genes. We have RH1, the rod opsin found in rods for dim light vision, and then 4 types of cone opsins found in different types of cones, each sensitive to a different portion of the light spectrum. And these are involved in bright light vision, as well as in some animals, color vision. Now in frogs and amphibians generally, they've lost the RH2 opsin, so they've lost one of those 4 cone opsins. And they actually, frogs and some salamanders are actually unique in having the SWS2 cone opsin expressed in a second type of rod. And so we think that this second type of rod enables frogs and perhaps some salamanders to detect color under dim light conditions when most other animals are colorblind, including ourselves. All right, so to look at this across taxa, we again took a fairly broad phylogenetic sample. Here we're going to focus on the different habitat types as well as the different developmental modes, the direct developers versus not. And we used eye transcriptome sequencing supplemented with whole genome sequencing, and then did molecular evolutionary analyses to measure selective pressure using what's called clade models. I'm not going to get into the details of these, just going to kind of highlight a few of the main results from here. And so, what we're measuring here is called omega. This is our estimate of selective pressure. And so in the rod opsin RH1, which is found in the regular, I would say, type of rod that typical vertebrates have, we actually see elevated omega in the scansorial species, so the tree climbing species. And we did a second analysis to determine whether this might be adaptive or due to relaxed selection, and that we don't find evidence that this is due to relaxed selection. So perhaps it's adaptive, and kind of our speculative hypothesis for this is that it may be involved in increasing the kinetic rates, so how fast a protein can respond to new light signals. And this could help frogs navigate these complex 3D environments, these scansorial frogs, under dim light conditions. We also found 2 of the cone opsins where we had elevated omega in aquatic taxa, so the LWS, which is the red rod opsin, as well as SWS1, which is the UV and violet, at least mostly in frogs, this is going to be violet sensitive opsin. And we think this is likely due to the differences in the light quality in water versus air. And we see a similar situation in fishes that transition between different water types. And finally, with SWS2, remember this is the cone opsin that's actually expressed in that second type of rod, we also see elevated omega here, so elevated selective pressure. And in this case, we do have evidence that it's due to relaxed selection. This is also one of the genes that we found was differentially expressed, at least in that one species we studied, between tadpoles and juveniles. And so what we think is going on here is that we have a release of selective constraint, because in these direct developers, this gene no longer needs to function in these 2 different light environments. And so we think that's what this evidence of relaxed selection is suggesting. Now, overall, we kind of conclude from this we have evidence, at least some preliminary evidence for visual adaptation, molecular visual adaptation in frogs based on their different ecologies and behaviors. But what are some of the next steps we'd like to take? We'd like to correlate these molecular results with spectral analyses where we actually measure the absorption of visual pigments. Now, we've done this for a small subset of our sample. And kind of what we could take away from this is that we do have considerable variation in absorbance spectra across these frog species, but there's no clear patterns right now. So we still have this really limited sampling though, and so really I think pulling out some patterns is going to require us to kind of finish this off with a larger sample of frogs. But we do find that there are kind of, to sum things up, some emerging patterns in frog visual evolution. And so we have scansorial frogs, we have evidence that they have higher eye and vision investment, as well as potential adaptations for higher visual acuity. Whereas we kind of see the opposite in the aquatic and fossorial species, where we have evidence for reduced eye and vision investment, changes that would likely result in lower visual acuity, and kind of overall suggesting a lower reliance on vision in these species. All right, thank you everyone for listening. A couple of my students will be presenting in the symposium coming up. One more talk on frog photoreceptor transduction evolution, and another student on turtles. So I just want to thank you for your time, and I'll take any questions if we have time, although perhaps not because we started late.
Right, so hi everyone. So my name is Benito Wainwright. I'm currently a research fellow based at the University of St Andrews, but the stuff I'm going to be speaking to you about today is stuff that I did during my PhD, which is at the University of Bristol, also in the UK. So if you look across planet Earth, we know that habitats that are more complex and the more heterogeneous tend to harbour more species, and that's because of a greater number of ecological dimensions by which species can partition themselves in these habitats and form niches. One of these dimensions is light, and in aquatic habitats, we know that species can adaptively partition themselves between different light environments that are created by, for example, these really dramatic depth gradients that you see in African Great Lakes. And this, in turn, can drive adaptive evolution of the visual systems within the animals that live in these environments. But we actually know relatively little about whether these same ecological and evolutionary processes can manifest themselves in a diurnal terrestrial environment, like a tropical rainforest, which are obviously very complex habitats, but differences between the light environments within these habitats is likely to be a lot subtler than in, say, an African Great Lake. Groups of neotropical butterfly are a really powerful model system for testing these hypotheses, in particular this tribe. So these are the Ithomini, they're the most diverse tribe of neotropical butterfly, where all the species within the tribe are unpalatable. And as a result of that unpalatability, species have converged in similar wing colours, morphologies, and behaviours to form these Müllerian mimicry rings within rainforests to better advertise to their predators that they're not good to eat. So this phylogeny on the left here contains all the species from our study site in eastern Ecuador. And as you can see, within a single community, there are these 8 different mimicry patterns, mimicry rings. And these are able to coexist because we know that they are segregated between different forest microhabitats. And that's driven by the ecological segregation of their predators, things like birds. But another thing we know a lot about these butterflies now is that they show really strong patterns of variation in visual system architecture, whether that be at the perceptual level, at the level of the eye, at the processing level in the brain, and even at the molecular level. So that suggests a high degree of evolutionary lability, but no one's actually ever measured the light environments in which these butterflies live and linked that to adaptive variation in their visual system. So what we did is we went to these forests and collected a bunch of spectral data and calibrated that spectral data for butterfly vision. And what we found is that this variation in ambient forest light can actually create a mosaic of microhabitat, which we correlated to a bunch of ecological variables. So on the y-axis here is photon catch, which is essentially the perceived brightness of the light to a butterfly. And hopefully you can see that that differs significantly depending on whether a spectral measurement was taken on a ridge rather than a valley. And we saw similar associations with loads of other ecological variables too, like height. So the higher you go in the canopy, the more light there is. So that validated to us that ecologically relevant light environments do vary within forest habitats. But what we then went on to show is that those ethomine mimicry rings that I showed you earlier are partitioned across these light microhabitats. So again, here we've got photon catch plotted against 2 other ecological variables, flight height and canopy openness, where each point is a different ethomine species and it's color-coded by its mimicry ring. And one general pattern that emerged from these data is that if you look at these ethomine mimicry rings, these kind of grey mimicry rings that have transparent wing patterns tend to be hunkered down in more shaded forest microhabitats, whereas those that have more flashy colours, these so-called tiger stripe patterns, are flying higher up in more open forest. So that told us that this abiotic variation in the light environment seems to have co-evolved with these biotic, mutualistic, mimetic interactions between species to maintain or maybe strengthen adaptive niche assemblages within tropical rainforests. But of course, then we were really interested to see as whether that can correlate with any visual traits. But one thing that's also very striking about ethomine butterflies is that they vary hugely in their wing morphology. And that presents a potential confounding factor because variation in wing morphology might translate to differences in flight performance, things like flight speed, which might potentially promote different visual adaptations completely irrespective of the light environment in which they're found in. So to get around that, we collected a bunch of wing morphological data and chucked that into a PCA with all our light and spectral data. And we found that this first PC axis here explained the majority of light environmental variation, and the second PC axis explains the majority of wing morphological variation. And hopefully you can see that if you look across that first light environmental axis, those clear wing and tiger stripe mimicry rings that I was talking about earlier segregate pretty nicely along that axis. And then there's this weird clear wing mimicry ring, which is aptly called confusa, which segregates along the wing morphological axis, which is why we treated that as a separate, what we call a mimetic cluster in our future analyses, because of its drastically different wing morphologies. So we then collected a bunch of visual traits. We started off by looking at peripheral light reception, the eye, basically. We collected a bunch of eye physiological data, which unfortunately I don't have time to show you. But we also looked at eye anatomy from dissected samples of eye cuticle. And we found significant positive associations between a bunch of these anatomical traits with that light environmental axis that I showed you earlier, in ways that cannot be explained by allometric effects. And one of those effects was in the overall number of facets, but another one was the average facet diameter, where species that live in brighter environments, in this case, seem to have convergently evolved wider facets, which is an interesting finding in itself. I'm happy to discuss that. So we then push further and kind of follow the trajectory of visual information to see whether variation in the light environment can also drive variation in the visual processing centers in the brain. So on the left here is a 3D reconstruction of a butterfly brain that was created from immunostained whole brain samples that had been scanned with a confocal microscope. And we used these 3D reconstructions to measure the volumes of different structures or neuropils within the brain. In particular, we were interested in these kind of orangey yellow bulbous structures either side of the central brain, because these are called the optic lobes, and they are the parts of the brain which process visual information coming from the eye. And again, press the wrong button. Again, we found significant positive associations with the relative investment in the optic lobe and the light environment. So here we've got the medulla and the lobular plate, which are just two of the neuropils found within the optic lobes. And generally, what we found is that species that have evolved to live in brighter light environments have convergently evolved larger optic lobe neuropils. So then we knew that these visual system and light environment associations existed. What we then wanted to finally test is whether convergence in mimicry alone can predict convergence in the visual system. And we tested that using a bunch of other statistical methods, which I definitely don't have time to show you. But the short answer is yes. So mimetic cluster can predict variation in the visual system, and these are non-allometric effects, which is evidence of adaptive shifts. But what some later evolutionary modeling confirmed to us is that co-mimics do seem to be attracted towards these convergent adaptive optima or adaptive peaks. So on the left here is what's called a phenogram. It's essentially a phylogeny of all the species within our community, but they're ordered by this trait, which is essentially a summary of all the visual and brain traits that we measured and that were relevant, and places where the lines cross is evidence of convergence. So hopefully you can kind of see that those mimetic clusters that I was talking about earlier are kind of all grouped together. So to conclude then, what we've shown is that light abundance within rainforests can contribute to multiple dimensions of adaptive niche separation and predict species distributions within these diverse tropical rainforest communities. But we've also shown that variation in the light environment can be a potent selective force within these habitats, predictably driving visual adaptations towards distinct adaptive peaks, whether that be at the peripheral and at the processing level. But perhaps the most exciting finding about all of this for me is that we're also showing what broad-reaching and perhaps surprising effects these biotic factors, in this case these interspecific mutualisms, have on organismal evolution. In this case, of course, the convergence in the visual system. So that's just about all I've got time for. I'd like to thank all these people, in particular my PhD supervisor, Dr. Stephen Montgomery, my two co-supervisors, Julien and Nick, and all these other people, in particular Theodora, who managed to put up with me for 2.5 months in the Ecuadorian Amazon. And thank you all for listening. Happy to take any questions. Yes.
You said you collected spectral data in the environment. Did you collect actual differences between wavelengths?
Yes, we did. Didn't have time to show that, but yeah. So what we did is we collected spectral irradiance and calibrated that to calculate the average, or the estimated photon catch of each of the different spectral photoreceptors using the butterfly visual model. And using that, we could estimate the relative abundance of different wavelengths. And the main finding we found from that is, yes, spectral differences do exist, but mainly in the long wavelength area of the spectrum. So these tiger stripe mimicry rings tend to be favoring habitats with a much more broader range of wavelengths in them. That's not to say that other parts of the spectrum don't vary as well. Like these Ethiopian butterflies are segregated at a pretty fine scale, actually. So all these butterflies that we sampled were caught within like 4 meters of the forest canopy. So presumably when you go higher, there's shift in the spectral composition of the shorter wavelengths as well. But yeah, it is happening. Yeah.
Sort of following up on that, I think John Endler showed back in the 90s that there's a really big change in the spectral shape of the radiance of the canopy actually closed like 90%. And I was sort of sitting here thinking like, well, is it 90% from my perspective or 90% from the butterfly perspective? And so, yeah, I mean, to what extent are there meaningful, like really big changes in the shape of the light?
Yeah, like it's difficult to compare those two studies, but yeah, so Endler measured basically the total amount of light that was available in the environment and didn't calibrate it for any particular visual system, at least in that paper. But the two different light environments which we see these clearwing and tigers, the light environments which they favor, seem to be analogous to the differences that Endler found, actually, when it comes to like small gaps and light gaps and all of that. But yeah, it's hard to directly compare them. But yeah, I hope that answers your question.
So, fantastic talk.
Thank you.
To what extent were you able to correct for phylogeny in these associations that you covered?
Yeah, I forgot to mention that actually. But yeah, all these analyses that we did were all done controlling for phylogeny. So there is a complete phylogeny of all the species within our community, our Ethiopian community. So yeah, all the analyses of the models were done using phylogenetic, generalized linear models and PGLS and all of that. So yeah, that was all controlled for. Yeah.
So, butterflies aren't looking at the butterfly wings. Somebody else is, right?
Sorry, I didn't quite catch that, sorry.
The color adaptation of the wings is not for other butterflies.
Mm-hmm.
It's for birds or something.
Mm-hmm.
What are the butterflies looking at? What is the important visual information for the butterflies?
Well, I mean, they're still using their wing patterns in mate choice. So that's pretty well established. But yeah, that whole area is something that I'm really interested in looking into a lot further, actually. Like the differences that we see, this light microhabitat segregation between the tigers and the clearwings, just, was just quite a sort of happy coincidence in the context of this study in a way, because, as you say, those colorations have probably predominantly evolved to signal to their predators. But something I'm really interested in looking at later is calibrating those same spectral measurements for bird vision and seeing whether that's actually driving the shifts that we see. But yeah, to cut a long story short, yeah, they are. They're using color as well to communicate with one another, and that's preferring particular light environments is probably adaptive to better signal to each other. All right. Oh, well, one more.
Do you know anything about the genetic basis of any of these traits that you studied and whether or not there's either some pleiotropy or maybe there's a chunk of DNA, some kind of supergene or something that's controlling all of the correlated traits?
No. Sorry. I mean, that would be really, really interesting to do. And kind of, yeah, link preference to genetic traits. But yeah, no, that's not my area at all, I'm afraid. But I would really be interested in looking at that, yeah. Okay, I feel like maybe someone else needs to come up now.
Hi everyone. Today I will talk about mosquitoes. I don't think I will introduce mosquitoes. That would be depressing. We know why we're studying mosquitoes. But let's talk about something way more fun, opsin genes, as we already talked for 2 talks. Opsin genes are proteins that are linked to a G protein that will trigger a cascade. And they are linked, they are binding a chromophore that is usually in the eye or retinal, which will be inside the protein and do interaction, non-covalent boundings with the proteins. All of this interaction will trigger the energy necessary for the chromophore to change its conformation and trigger the cellular cascade. So basically, each time you have an opsin protein, the sequence of amino acids will define the energy necessary to trigger the cellular cascade, and so you can see different wavelengths. So usually, the more you have opsin genes, the more you are able to discriminate colors if you have the right neuronal interactions. And ancestrally, in insects, we have 3 opsin genes. One which will be sensitive to UV, another one to blue, and another one to long wavelengths that would be either green or red. And the amino acid sequence will define the spectral sensitivity. So why mosquitoes? In mosquitoes, you have a huge diversification of these opsin genes. They have, in average, around 10 copies of these genes. For an example, a comparison, in butterflies, 10 is just like an exception, it's outliers. Usually we are around 4 or 5. And in flies, these opsin genes can bind something else than the classic retinal chromophore. And so they can have other functions that can be heat sensitive, they can be mechanosensitive, or they can act as receptors for chemicals. So in mosquitoes, only the females will bite. And so we have this dimorphism between a male who will just go and forage flowers and a female who will look for blood. And in the female, if we knock out some of these opsin genes, we will remove the preference for skin in mosquitoes. So our question is, which opsin genes are involved in vision and what are opsin genes involved in? For that, we are using a combination of 3 approaches. We are doing bulk RNA-seq in the eye of the mosquitoes, differences between male and female, to know which one are involved in vision, and are they the same in males and females, or do we have any differences? Do they see the same or not? Then after, we combine that with microscopy to see the pattern of expression of these opsin genes across the retina. And finally, we did single-cell RNA-seq on the full head of the mosquitoes to see where are expressed opsin genes in the different cell types of the head. So to do this analysis, we are using 3 different species. Why these species? First, because they are available in the lab, but also we took 2 biting species, one diurnal, one nocturnal, and a non-biting species, which is diurnal, which is Toxorhynchitas buripalpis, which will act as a control to know which genes are involved in biting or not. And all of them are quite a lot of opsin genes. So when we do the bulk RNA-seq, we did that in standard condition, whatever. What we have is 3 species, and we look at the differences between male and female. So what we can expect is that all of the differences that are common between the non-biting species and the biting species will be related to sexual differences. However, all of the differences that are shared between the 2 biting species that we have will be related to biting processes and the biting behavior. So what we did is that we did bulk RNA-seq in the 3 species. For now, I'm struggling with the analysis of the Aedes, so I will not give you a full picture of that. However, we did it in the non-biting species and Anopheles gambiae, which is vector of malaria. And what we found already is 8 genes that are differentially expressed between male and female in the eye of both of the species. So these are the different genes, and basically I will not go in details on what they are because these differences are between male and female. They are not opsin genes. They are not about the vision. However, we can see that we have 5 genes that have the same differences in the 2 different species. So because we don't have a lot of annotation in the non-biting species, I will focus here on the Anopheles gambier analysis. We have 234 genes that are differentially expressed in the eye between male and female. And when we look at what are these genes, they are not opsin genes. We have a lot of visual-related genes that are differentially expressed. But more interestingly, if we do a GOTEM enrichment analysis, we pick up sensory system and odorant receptor differences between male and female. And in total, we have 10 odorant receptors that are differentially expressed between male and female in the eye. And only a single one for now has been found also differentially expressed in the non-biting species. So then after, what about the opsin genes? When we look at the opsin genes, this is the list of all of the opsin genes in Anopheles gambier. The red square will define the ones that are significantly differentially expressed, but the log fold change is very tiny. So the differences between expression between male and female are very small. So what we thought is that maybe there are differences of expression because they are tiny that maybe are in the pattern of expression among the retina. So to do that, we did some microscopy. This is just my quota of pretty images because mosquitoes are not very pretty. So what we find basically on the right image, you have the retina, the full retina of the mosquito, and we find a stripe. On the left, I put omatidia of mosquitoes. An omatidia in the mosquitoes, it's 8 photoreceptor cells, 7 that are around the radome, and 1 on the bottom. And basically, what is happening there is that there's only one photoreceptor cell that change and do the stripe. It's R7 that will either express blue or UV. All of the ones around will co-express 5 different long wavelength opsin genes. And so what we thought is that maybe we have a difference in the proportion of the stripe between male and female. We did some quick analysis with a few number of individuals. What we see for now is a trend that the dorsal blue stripe is smaller in females compared to males, but this is not significant until now. So if we compare what we know from the literature in the other biting species, we see that the structure of the retina and the expression of the opsin genes across the retina is completely different. We have something which is complex in both cases with stripes in both Anopheles and Aedes. However, in Aedes, all of the opsin genes are always co-expressed. That means that you will never have only a blue or UV. There will either be blue and long wavelength or blue UV and long wavelength, which means that probably they have less discrimination of the colors compared to Anopheles gambier. So then after what we wanted to do, until now, we just show bulk RNA-seq, so we just average the expression of the genes inside the tissue or the eye. So we wanted to go single cell level. So we can, by doing single cell, have the mean expression of all of the different cell types that are in the head of the mosquitoes and compare male, female, and between species. And all of these different cell types can be neurons, odorant receptor cell, glial cell, photoreceptor cells, whatever is inside the head of the mosquitoes. So we did these experiments in Anopheles gambier. When I arrived, my PI told me, "You are French, so just cut the heads." So I just cut the heads. And I did a lot of them to do multiple replicates for that. This will be preliminary results because we are doubling the sequences to have a better resolution. But basically what you add out of it is a clustering. So each color will be a cell type and each dot will be a cell. And we can assign which cell type is what. So we easily find the photoreceptor that are in green on the left. And if we look at the expression of the different opsin genes in the different clusters, what we find is the main expression in photoreceptor cells, but also expression of especially the 3 first ones that are long wavelengths opsin genes all around the head of the mosquitoes. And I was lucky that some collaborators in Rockefeller University did the same single-cell atlases in the other biting species, which is Aedes aegypti, which is a vector of the yellow fever. And the only thing that I want to point out is this square, which is one long wavelength opsin genes, which is mainly expressed in photoreceptor cells, but has a high expression in a lot of different cells, including odorant receptor cells. And guess who it is? It's the one that if you knock it out, it will remove the preference for human skin. So as a conclusion, this is like a bit like not really good, but until now, when a mosquito is trying to find a host or whatever, interacting in an environment, you don't use only one cue, but you use a plurality of cues, and all of these cues has to be integrated together to have the behavioral response. Until now, we often thought that all of this integration was happening in mushroom bodies or central brain. What we saw is that opsin genes are already expressed in odorant receptor cells, and some odorant receptors proteins are already expressed in the eye of mosquitoes. So this interaction is probably way stronger than what we thought, and the integration is probably happening way before what we thought until now. And secondly, opsin genes are often studied just for the visual part of it, but here we show that opsin genes are expressed all around and can have a lot of different functions. And on that, I will just acknowledge my lab and all of the collaborators. Thank you.
For this opsin gene that's involved in detecting skin, are there any changes at the amino acid level that you think could kind of indicate potential changes in function?
So the problem is that basically opsin genes in mosquitoes are a mess because you have a lot of duplication, and so we don't know which one is which one in the different species. So it's hard to reconstruct what happened and which mutation had an impact on the function of this specific one. These are current work that we are doing. We're trying to do more phylogenomics to try to understand what happened and see where are the mutations happening and under which selective pressure they are.